Today's video topic is light reaction of photosynthesis. So what is light reaction? It is a photochemical phase which include light absorption, water splitting, oxygen release, and formation of ATP and NADPH. So basically in light reaction, ATP and NADPH is formed which later utilized in dark reaction in stroma for formation of glucose. Okay. So light reaction take place in thylakoid of chloroplast. So suppose that here is a chloroplast. Okay. Inside the chloroplast, a coin-like structure present that is called thylakoid. Okay. The space inside the thylakoid is called lumen and the space outside the thylakoid is called stroma. And thylakoid have a single membrane which is made up of lipid bilayer. Okay. So for formation of ATP, the concentration of H ion, there should be a concentration gradient of H ion between the stroma and lumen. In lumen, the higher concentration, and in stroma, the lower concentration. Okay. So for that, there is two photosystem present in thylakoid membrane, photosystem 2 and photosystem 1. So first light fall upon photosystem 2, okay. So electron get excited and jump to the higher orbit and transfer to the plastoquinone. And then plastoquinone transfer this electron to cytochrome B6F and cytochrome B6F transfer this electron to plastocyanin. Okay. So this transfer of electron from PS2 to PQ, B6F and PC is called electron transport chain. Because PS2 is keep transferring the electron, it is also losing the electron. Okay. So for replacing those lost electron, the water is split inside the lumen. This is called photolysis of water and it releases hydrogen, oxygen and electron. Okay. This electron is supplied to the PS2 and the oxygen which is released, that is released in stroma. Okay. And it go outside the stroma and released in environment. Hydrogen ion which is released, it increase the hydrogen ion concentration inside the lumen. Okay. Now the PQ, okay, this PQ grab the H ion from stroma, okay, and transfer that to the cytochrome and cytochrome transfer that H ion to the lumen. This also increase the concentration of H ion inside the lumen. Okay. So in both way, by splitting of water inside the lumen and by transferring the hydrogen ion from cytochrome, the H ion concentration is increased inside the lumen. Okay. Now, because PQ is using the stroma H ion and sending it to the lumen, it decrease the concentration inside the stroma. Okay. So PS1, okay, this is done by PS2. Now PS1 also absorb the light, electron get excited and go to higher orbit and it transfer to the ferredoxin. Ferredoxin is a NADP reductase enzyme, okay, which utilize this electron and combine NADP and hydrogen to form NADPH, okay. Because PS1 is also losing the electron, for replacing those electron, plastocyanin transfer it electron to PS1. Okay. So PS1 is getting electron from PC and PS2 is replacing the lost electron from splitting of water. Okay. Done. So here NADPH is produced and because the H from stroma is utilized in the formation of NADPH, it also decrease the concentration inside the stroma. Okay. So in this way, the concentration gradient of H ion build up between the lumen and stroma. So when this concentration gradient is built up, the hydrogen ion passes through a protein channel. This protein channel have two parts, F node and F1. Okay, F node passes the hydrogen ion quickly across the membrane, and F1, when the hydrogen ion passes rapidly, it generates energy. F1 ion is an enzyme, ATP synthase. This F1 uses this energy and combine ADP and phosphate and form ATP. Okay, so that's how ATP is produced and NADPH is produced. Both ATP and NADPH is produced at the outside membrane of thylakoid and released in stroma. Okay. So what we learn here, that in light reaction, what take place? Light absorption. So PS2 and PS1 both absorb different wavelength of light. Done. Water splitting. So for replacing the lost electron in PS2, water split inside the thylakoid lumen. That is called photolysis of water, and it releases electron, hydrogen, and oxygen. Oxygen release. So by water splitting, the oxygen is released. That oxygen go out of the stroma to the environment. Formation of ATP and NADPH, that also took place at thylakoid membrane. Okay, so all these events together called light reaction because it take place in the presence of light, or here you can say the light is involved directly. Okay. Now, in this process, you can see that both PS2 and PS1 is involved. Also, the electron which is transferring from PS2, okay, through redox chain or electron transport chain, is not coming back to the PS2. Okay, so it is called non-cyclic phosphorylation. Okay, because phosphorylation means ATP is produced. Okay, this is ATP formation is called phosphorylation, but in this process electron is not coming back to its original source. Okay, so it is called non-cyclic. What is cyclic phosphorylation? That take place in stroma lamellae. Okay, so in stroma lamellae, only one photosystem is present. So this photosystem absorbs sunlight, and what happened? Chlorophyll A get excited, electron get excited, jump to the higher orbit, and then it transfer to the ferredoxin, then PQ, cytochrome, then PC. Okay, ATP is produced, but electron come back to its PS1. Okay, so it is called cyclic photophosphorylation. And also here the ADP is formed for the supplying the extra demand of ATP production inside the cell. Also in this process, water doesn't break down, oxygen is not released, and NADPH is not produced. Okay, so this is about cyclic and non-cyclic phosphorylation. Also, there is one question, what is JED scheme? Okay, so see, in thylakoid membrane, during the light reaction, it is PS2 and it is PS1. Okay, PS1. So when sunlight fall, electron get excited. Okay, so it go to the higher energy orbit. Okay, then it loses the energy, transfer to PQ, again lose the energy, transfer to cytochrome, then PC, and then go to PS1 and again absorb the light and jump to the higher orbit. So here energy is going up, down, up. This form a JED-like structure. This is called JED scheme. Okay. So the topic is done. I hope you like it. Please like, share, subscribe my channel. Thank you.